Do spiders have friends? It may be a question that's never crossed your mind at all, but sociobiologists have been studying variants of it for a while now. Research on social spider species has been alive since at least the 60s. Let's first talk about what sociality is, and then let's talk about an Australian example shown on the screen here, Anelosimus pratchetti. Sociality exists on a spectrum. Broadly, it ranges from being entirely solitary to entirely social. Spiders exist along the breadth of the scale. Many huntsmen are solitary with minimal parental care. My study species, Anelosimus pratchetti, falls into the range of subsocial. As for social, Anelosimus makes an appearance in Anelosimus studiosus. Finally, some animals are eusocial, like ants and bees, and some researchers even place Anelosimus eximius into this category, although that's not universally supported. Anelosimus species in particular are known to have evolved varying degrees of sociality independently of each other, and have the highest number of social species of any genus. Here's a phylogeny of the Australasian Anelosimus. There are several species that have not been studied in depth, but many display sociality of some form. However, the knowledge for a lot of the non-American Anelosimus is very shaky, with the evidence for Anelosimus pratchetti boiling down to some researcher watching them for a while. You're currently looking at the entirety of published literature on Anelosimus pratchetti down there in the bottom left. Congrats, you're an expert. Needless to say, this is the knowledge gap I was seeking to fill. I opted for an obvious and basic question. Are these spiders able to recognize family? This will help us understand if this species is present in distinct nest units, where the spiders from the same location are part of some supernest, or even if all spiders of the species are just buddies and get along well regardless of their origin. While we're sadly unable to take a field study of the many Anelosimus species to determine the origins of sociality, Agnarsson, 2012, makes the point that observing distinct lineages of Anelosimus and using them as evolutionary replicates is important to pick apart the ecological factors that contribute to the evolution of sociality. This motivates an investigation of the sociality of our very own Anelosimus pratchetti. Hopefully, it yields insights that slot in with the bigger picture of social evolution. Conveniently, Anelosimus pratchetti is present in bulk in and around Smiths Lake Field Station. We found colonies along the Treachery Head walking track down at Seal Rocks, and I found a large colony around the field station as well. Colonies range from 0 to 35 spiders, with a mix of juveniles, males, and females, indicative of at least subsociality. There was also an abundance of kleptoparasites and possible predators present within the webs when we pulled them apart. As you can see on the map, these locations are quite distant, about 6 kilometers as the crow flies and about 11 kilometers when following roads and trails. After dissecting the nests, we annexed the rear of the food hall and repurposed it into a spooky dungeon lit only by red light, achieved by taping bread paper over the lamps to simulate nocturnal conditions where the spiders would be most active. Spiders were then left in these conditions overnight. Water was available through a moist paper towel, and the lids, also paper towel, were breathable. After about 24 hours of equilibrating to the spooky conditions, I set up my treatments. I placed my spiders into Tupperware containers, using 3 treatments of spiders from the same colony and 3 of spiders from different colonies. Water was available to get through a moist paper towel, placed in the center, and air holes were poked in the lids of the container. Herding the spiders into the appropriate starting positions was difficult, but I managed it. 7 spiders were placed at either the north or south ends of the container, making 14 total spiders. The lids were then not opened for the rest of the experiment to avoid web disturbance. This was my setup. Hourly for 30 hours, I checked and recorded the clustering of spiders. Here's a picture I took at the end of the experiment. Pause and observe the clusters if you like. Sadly, I was unable to mark spiders individually since due to the red light conditions, paint dots would have been impossible to see, and also painted spiders seem to change their behavior when marked. I got around this by placing spiders in groups at opposite ends of the containers after making sure they were capable of traversing the container. Spiders therefore had a choice, stay with their original group or mix with another group. At about 3 in the morning, an intruder got into my setup, but thankfully he was friendly and he left the spiders alone. The data that I collected ties into an idea of E.O. Wilson's. If groups are permeable, spiders form large clusters and mix freely between them, then there's likely to be high gene flow between them. If groups are less permeable, there's less likely to be gene flow. While social species can have both high or low permeability in mixed colonies implies that spiders can recognize members of a different colony. This also implies a degree of inbreeding within a colony, a trait considered to be on the road to eusociality, similar to termites, ants, and bees. By comparing the different containers over time and looking for differences between their clustering, it will tell me whether mixed colonies have different permeabilities. I predicted that mixed colonies would have lower permeability than my controls. If spiders displayed the same behaviors in my control and my treatments, I wouldn't be able to definitively conclude whether the spiders can't recognize each other or if they just don't care. This would be an interesting result, and could contribute some explanation as to why these spiders tolerate kleptoparasites in their webs. More broadly, this work contributes to examining an evolutionary replicate in the Anelosimus genus, just another brick in the wall of knowledge about sociality in this fascinating genus.